Welcome to Lantern Pharma interview session. Today we are joined with a special guest, Dr. Reggie, Vice President of the Clinical Development and at Lantern Pharma. Dr. Reggie, you are welcome.
Thank you very much for having me here to talk about Lantern Pharma and our recent data.
Thank you for joining us today. In this session, we will dive into promising results from the HARMONIC Phase 2 trial, focusing on LP-300, its significant implication for treating lung cancer in never smoker. Additionally, we will explore the transformative role of artificial intelligence in drug discovery, particularly in its responsive clinical trial and treatment strategy. Could you start by sharing some information about your professional journey and mission of the Lantern Pharma?
Thank you much for that first question. I think it should come as no new information to the audience that one of the major tools in the treatment of diseases is obviously drugs. My professional training as a physician is in the areas of clinical pharmacology and oncology. Now, the intersection of both specialties made it possible for me to develop a unique insight into how cancer drugs could be developed more efficiently and used in the clinic. So after a brief stint as a faculty at Ohio State University, I joined the pharmaceutical industry and have been since involved in the development of several anti-cancer drugs for well over 2 decades. Currently, I lead the clinical development program at Lantern Pharma. Now, your other part of the question was the mission of Lantern Pharma. I think this is very simple. Lantern Pharma is leveraging artificial intelligence, machine learning, and genomics to transform the costs, the pace, and the timeline of oncology drug discovery and development. And we have several examples in our pipeline where we've been able to cut down timelines and costs, which is unheard of in the industry as we look at our assets, getting into the clinic.
The smoking has traditionally been recognized as a major risk factor for lung cancer. However, there is a growing group of lung cancer patients who never smoked, presenting a new challenge for treatment. Could you provide us with an overview of the current statistics and trends for both groups?
The unfortunate thing, though, about lung cancer is that it remains one of the most common and deadly cancers worldwide. If I stick with the United States, for example, the American Cancer Society estimates that in this year alone, about 234,580 new cases of lung cancer, which unfortunately in more women than men will be diagnosed in the United States. And about more than half of those, 25,070 deaths will result from lung cancer. So it is really common but deadly. Now, the most common type of lung cancer is what is called the non-small cell lung cancer. This represents about 85% of all lung cancer. And then there's a subtype of it, which will be relevant to our conversation later on, is the adenocarcinoma. Is the most common subtype of lung cancer. Now, although most people who develop lung cancer have a history of tobacco smoking, about 10 to 20% of lung cancer cases occur in people globally who have never smoked. Now, that number is much higher, more than double in the Asia Pacific region, but in Europe and the United States, it's about 10 to 20%. Now, lung cancer in never smokers occurs more frequently in women and also at an earlier age than in smokers. Now, there are 3 differences I really want to emphasize between these 2 groups, the never smokers and the smokers. Most tumors in never smokers are adenocarcinoma, and they arise from the accumulation of mutations caused by natural processes in the body. These tumors tend to have distinct molecular characteristics, and their evolutionary process is also different. which is kind of interesting and intriguing at the same time, but it creates an opportunity for drug development for that subpopulation. Now, for the smokers, on the other hand, they tend to have tumors that have multiple mutations, or what you call increased tumor mutational burden, and predominantly develop a subtype called squamous non-small cell lung cancer and small cell lung cancer. The second difference between these two groups is that never smokers do not tend to derive clinical benefit from currently approved immunotherapies, which is sad because that is really one of the frontiers right now, in cancer treatment, the use of immunotherapy, but these patients don't derive benefit. And lastly, while lung cancers in smokers is on a gradual decline, actually, because of all the preventive measures, stop smoking, all those public health measures, that is not the case with never smokers. The never smoker population or portion of lung cancer patients is really on the increase. Thus, the need for development of therapies that will significantly benefit never smokers with lung cancer.
In the HARMONIC Phase 2 trial, you reported an 86 clinical benefit rate in the initial patient cohort for LP-300, and the objective response rate stands at 43 with a notable reduction in tumor size. This is very important figure. Could you analyze this result in comparison to existing treatment and to discuss its potential impact on the future clinical practice?
Well, I think I want to put that data in context. These are preliminary data, and much later I will explain the study design. But nevertheless, it's always very exciting when you do drug development in oncology and you begin to look at what potentially might be predictive of the drug treatment, the drug itself, or the regimen. Now, there are three elements. We have the benefit rate we talked about of 86%, which literally means the majority of the patients in the study are benefiting one way or the other. They have not progressed while on treatment. The second is those who have a quantification of their disease reduction to a level you call response in terms of partial response or complete response. Now, it is customary to evaluate clinical benefit in oncology drug development with three considerations in mind. So as I talk about the data, I want you to bear that in mind. There's one of the onset of the benefit. Now it's traditional for us to use what we call a spider plot to look at that data. That looks at the onset, how quickly are the patients responding. The second is the magnitude of the benefit. We use a waterfall plot to look at that. And the third, obviously, is the durability of the benefit, which is we use the swimmer's plot for that. So looking at those three characteristics individually on this preliminary set of data, a look at the spider plot itself demonstrates two emerging encouraging attributes of the regimen. The onset of clinical benefit is rapid. So it's not delayed. It's something you see very quickly. And it will appear from that same plot that this is looking durable. For example, the first patient that have derived benefit in the study, which was the second patient enrolled in the study, is on now for more than a year in that study and has maintained the response. And is looking a lot better, obviously, in terms of his daily activities of living and what have you. So that's the spider plot. We see that in the patients that have responded or are deriving clinical benefit. It's rapid. Now, with the waterfall plot, the magnitude is also very impressive. And the response just after 3 cycles of treatment, each cycle is every 21 days. So basically, after 3 administration of the drug in combination with the standard of care, virtually all patients, we saw that impressive magnitude. And again, I will speak about the demographics and the specifics that we saw in the data much later on. Now, the third aspect is the durability, which we are seeing with the swimmer's plot, and we are monitoring that. But at least, at the minimum, we've observed all patients who were valuable for response assessment after 3 cycles have continued to derive clinical benefit thereafter. And there have been no drug discontinuations due to toxicity, which again is something not common in oncology drug development. So they've stayed on their treatment since when they started treatment. There's no reduction, no interruption. They've all stayed on that. The other thing that is very telling in the data is that the median number of prior lines of therapy, it ranges from about 1 to 4 in this group that we've seen so far. Nevertheless, the data suggests that regardless whether they've had 1 line before or 4 lines of prior treatment, or the type of tyrosine kinase inhibitors that they failed, using LP-300 in combination with standard of care regimen is likely to provide clinical benefit. Now, it's important to note that one of the patients that we've seen so far had already been exposed to this standard of care treatment, which is the carboplatin plus pemetrexate, but still derive remarkable benefit. Actually, is the one that's been on the longest, in the study, which suggests to us that this regimen actually has a very strong potential in this space.
Well, moving the next question, and could you explain how the LP-300 mechanism of action interact with the tyrosine kinase inhibitor and affect the cancer cell in never smokers?
Yes, thank you for that question, because LP-300 actually has been in development prior to this particular study. Initially, when the drug was being developed, there was an understood mechanism of action that did not include the understanding of its effects on the tyrosine kinase receptors. And I will talk about that. So it has, now we know, 2 broad mechanisms of action. One is that it is known to resensitize tumor cells to chemotherapeutic agents by resetting the redox cycle. This was the prevailing thought in terms of its mechanism of action in previous combination regimens that were used. However, in 2 of those pivotal trials or later stage studies that were done, we recognize that patients that are never smokers literally double their benefit compared to those who did not get LP-300. So that raised the question, what is it about LP-300 beyond this resetting of the redox cycle that seemed to benefit this subset of patients? So we now know, and also based on further preclinical studies that we've done, that actually this drug, when you look at never smokers, they tend to have a higher percentage of mutations in tyrosine kinase oncogenes, such as EGFR, ALK, ROS, MET, to mention, and the majority of those that contribute to the tumor growth and formation. Now, such tumors in smokers are much more likely to have growth-driven mutations in oncogenes such as RAS, and not these tyrosine kinase receptors that I've talked about. Now, we figured out much later from preclinical studies that actually the drug, what it binds to an area of the tyrosine kinase receptor, and by doing that, it really impacts how those receptors transmit signals, for the disease to continue and so on. So we now know that clearly this drug actually is a multi-tyrosine kinase receptor inhibitor, so to speak. So, and you will see that in our patient population and in the study design, what we've done is not to restrict it to one particular mutation, but to literally say, regardless of the mutation that the patient has, they will potentially benefit from the drug. And we are beginning to see that actually in this early set of data. The patients all are not in one box. They have different type of mutations, sometimes co-mutations, and yet they are deriving clinical benefit, which begins to validate in some extent our thought around the mechanism of action of LP-300.
How do patient demographic and tumor mutation burden influence the efficacy of the LP-300? And what does this mean for personalizing treatment approach in lung cancer therapy?
Yeah, that's a very great question. And one for which, obviously, as you develop a drug in oncology, we try to look at what the data is telling us and to kind of adjust. And after all, we are an AI company. So we obviously listen to the data very early. We interrogate it as much as we can so that we begin to formulate, as it were, some algorithm or where the data is leading us. Now, regarding demographics, there are a couple of encouraging observations specific to tumor response in this early data set that we're seeing. Now, one of that is that the patient population is relatively more heterogeneous. That will not be the case with typical studies in this space, because what tends to happen is you have a particular drug is going after a particular target, and then patients that are allowed in that study tend to have just that one driver mutation. That is not the case with our patient population, of course, based on the scientific rationale. Now, also, the tyrosine kinase receptor genomic alterations at baseline that we've seen in our patient population is other than EGFR or EGFR alone. We've had a patient with co-mutation. We've had more than just EGFR, again, which speaks to what we believe about the mechanism of action and the potential, benefit or the potential value that this drug might bring into that space. The tumor response is, interestingly, is exaggerated at the metastatic lesion sites as we look at this patient. So if we look at baseline scans and we look at scans once we evaluate this patient, those that have clear metastatic lesions, they either disappear completely or there's resolution to normalcy in the size in case of lymph nodes. So again, these are all very encouraging signs to us. Now, to answer your question around the personalization of treatment, this also is very intriguing, because what we've observed in the data set is that the patients with low or intermediate tumor mutational burden are deriving clinical benefit. Now, we believe in the era of personalized medicine that this will pave the way for a more acceptable and adaptable biomarker selection strategy, since the tumor mutational burden is already part of routine clinical practice. So overall, the take home for us here is that tumor mutational burden might end up being really one of those personalized medicine approach. Is easily available and practiced right now in terms of stratifying patients. So we are right in the middle of that. We don't have to invent anything new, but at least this is data that is encouraging to us, at this point.
What do you believe the impact of your finding from the HARMONIC trial will be on the future of the treatment of the lung cancer?
In a very simple way of putting it, which I try to do, the impact will be very significant and most welcome. If one takes a look just at benchmark data, when pemetrexed and carboplatin was evaluated in similar population, the response rates across the board have ranged between 20 and 30%. In this preliminary set of data that we have, we're already observing a response rate almost double that, right, 43%. But more importantly, the median progression-free survival in those historical studies, even some that are fairly recent, the median PFS is about 4 to 5.5 months, even in combination with immune checkpoint inhibitors. Now, this is considered suboptimal. Now, these are very early for us, so we really haven't estimated the median PFS. But nevertheless, on the trajectory that we are seeing currently with this set of patients, I think, is kind of encouraging, what we're seeing. And we believe very strongly that the combination will ultimately be very helpful in this population. Now, let me also state this, that the combination regimen currently approved with a bispecific, in speaking with investigators that are participating in the HARMONIC trial, they're already expressing concerns about which patients are more likely to benefit given the toxicity burdens, of that regimen. As an example, patients that take that regimen, about 34% or more of them are going to discontinue one or more of those treatments. And the majority of them end up having real, significant adverse events. So we believe in a space that, looking at the data we have currently, this is really, really very competitive. If the current observed clinical benefit profile stays the same, with more patients enrolled into the trial, undoubtedly, this will have a significant role in the future treatment of never smokers with lung adenocarcinoma. And as I stated earlier on, patients with low and intermediate TMB are deriving clinical benefit. And if this trend holds up, this also will pave the way for an adaptable biomarker selection strategy, since this assessment is already part of routine medical care. So overall, we're very confident. This obviously, if it holds up as we think it will be a significant improvement in terms of the treatment available for this patient population.
And what about the side effect? And were there any safety concern observed during the trial? And how do this compare to the typical conventional treatments?
I will answer that again with a very brief, simple statement. There have been no safety concerns. Let me just explain the way the study is designed. I'll use this opportunity to do that. The HARMONIC study includes two stages to it. The safety leading, which really relates to what you're asking now, and the randomization and expansion stage. Now, the design is completely aligned with current dosing interval for the standard of care regimen. Now, I say that so that we'll put that at the backdrop and in context with the safety profile we've observed. Now, in this safety leading so far, the regulators, the FDA, required us to really ask the question, what is the safety profile when we use LP-300 in combination with the standard of care regimen, which is pemetrexed and carboplatin? We've presented the data since we've completed that stage with the Data Safety Management Board. So they've looked at it, the monitoring board, sorry. They've looked at it, and unanimously, even investigators as well, have concluded that the events we've observed, the safety profile is in keeping with what is expected of the standard of care regimen. Overall, just looking at the safety profile, we observe primarily grade 1 and grade 2 toxicities, and most of these were really attributable to just one patient in the study who had a lot of comorbidities coming on. But there's no doubt that if you look at the safety profile so far, there's nothing unexpected. There's no new toxicity, which is important. But more importantly, as I mentioned before, patients on the 3-drug regimen have continued telling us that this is tolerable. So it's not just safe, as we say in oncology drug development, but how tolerable is the treatment? And yeah, this is tolerable because there are no discontinuations of LP-300 or so. So clearly, there have been no safety concerns, and we are really, really encouraged. And we moved on now to the randomization stage.
Looking beyond current trial, what are the next steps for Lantern Pharma in exploring expanding the therapeutic potential of the LP-300?
Yes, as I mentioned, the trial right now is in patients that have already failed at least their first line of treatment with a tyrosine kinase inhibitor. Now, based on the mechanism of action and this emerging clinical experience in the trial, we're already in discussions with investigators, evaluating opportunities and study designs that will expand the therapeutic use of the drug into earlier lines of treatment. This is routine. This is usually what is done. And we are very, very hopeful and excited actually to evaluate those opportunities and interrogate the drug in earlier lines of setting. But certainly we want to see how this new set of data matures. I think that will also give a lot of credence to the fact that and comfort for investigators to use the drug in earlier lines of setting rather than where we are, past first line of treatment.
So the last question, which is very important also for this study and the future of the drug development. As AI continue to play a critical role in the drug development, how do you envision the future of the integration of the artificial intelligence and machine learning in clinical practice and particularly for the oncologist, what are the next steps?
That's a very, very intriguing and complex question, because you bring in the components of clinical practice into it. As I talked about in my introduction, I obviously was in practice for a long time before getting into industry. So I have this privilege of really being on both sides and trying to figure out, certainly in Lantern, what would be the role of AI and machine learning in clinical practice. But I think one might want to take a step back and ask the question, in drug development, because before you get to practice, you really have to ask the question on the front end so that you provide the appropriate tools, to the oncologist, those of us who develop the drug. So with that in mind, Lantern Pharma has a platform called Radar, and it's used primarily to predict the potential response patients will have to Lantern's drug now. That's what we're doing right now. And to other drugs that are being reviewed and analyzed by Lantern. Now, I want to emphasize that because we start like in a dish, and then you continue to expand that. So we are also, the radar is also being used to help define and develop combination strategies among drugs in development and those that are approved for a range of oncology indications. So in terms of clinical practice, just right there, one of the things we're doing, there are a lot of drugs being used in practice right now. And the reality is, in a lot of cases, there's really no added information as to how best to repurpose some of those drugs or rescue them, those that have failed. But through our Radar platform, we use transcriptome data, genomic data, and drug sensitivity data from a wide range of curated sources that are continually being analyzed, monitored, and updated. Now, through that platform, which is really our core approach, to drug development, we identify candidates for in-licensing and development, but also in collaboration with other sponsors, we help them to figure out a development strategy, either in combination or in figuring out patients that are more likely to respond, to drugs. Now we have surpassed 60 billion data points in this effort, facilitating increased drug and cancer type specific biomarker identification, the discovery of new indications, and the identification of additional drug candidates to build out our product pipeline towards advances in cancer therapy. So if I look at your question around what are the next steps, I think it comes back to what I started with, which is that at some point, stakeholders, including oncologists, right, the education of stakeholders, that will be critical. And the deployment and or integration of the tool in some form or shape beyond drug discovery, but into clinical practice. And there are currently certain aspects that could be of immediate value. For example, we are repositioning already approved drugs, especially in combination regimen as applicable. Now, these are all efforts we're doing. Of course, we have publications on our website with a particular drug called spironolactone. We published on that. This is a drug being used for treatment of, hypertension and other endocrine diseases. But actually, we've now found that you can reposition the drug in combination with one of our assets to make the asset better. But it's a personalized approach. So again, these are some of the efforts, and things we're doing with AI.
Thank you very much to sharing this important information and sharing your insight and expertise with us. It was a pleasure for us to have you in this meeting.
Thank you very much for having me.
And thank you very much to everyone who joined to us for this webinar. Please share your question, thought with us by writing in the comment section. We are looking to welcome you in our future events. Have a wonderful day.